Welcome back to Learning Image Analysis. Today we'll be covering how to characterize an image by multiple segmentations. So here's our image here. It's a neuron-like cell, technically a PC12 cell, that has been stained fluorescently, first with green, which stains the cytoskeleton. Then we have red, which stains the mitochondria inside the cell. Then we have blue staining the nucleus. So the research question that we're asking about this cell is where the mitochondria are located. Are they here in the cell body, or have they been transported out to the neurites here, these extensions? So we want to characterize this as a percentage, the amount of mitochondria in the neurites versus the percentage of mitochondria in the cell body. For our learning objectives, first, measuring one segmentation relative to another. And we'll also talk about using different segmentations and combining, again, the information from them to partition the mitochondria into the cell body versus the neurite. We'll talk a little bit about understanding needed segmentation accuracy. How well do we have to have the cell body defined in order to be able to answer our question? We'll discuss potential variation between your images. And lastly, deciding the best way to measure a feature. Now, looking at our image, the first thing that we notice is that there's a scale bar here in the lower left. And of course, we want to get rid of that. Now, depending on the subsequent steps that we do, determines the way we want to remove the scale bar. If you remember from some past lessons that we did, we can either fill the scale bar in with black, or we can segment the scale bar and then subtract it from subsequent segmentations. And which one you want to do depends on the type of processing that you'll be doing later. Is it important to have the actual grayscale values removed or not? We'll start with the simplest method at first, just getting a segmentation of the scale bar and then subtracting that. And if it turns out having these white pixels here in the image is an issue, we'll fix that later. So if we start with a simple basic threshold here, right, let's see what values are good. Now 254, here's our threshold. It tells us the scale bar isn't solid white like it's supposed to be. You can see some speckling over here, and that's because of the JPEG compression that we have going on. That's what causes it to not be uniform. So let's bring this value down a little bit to 45, 240, okay, 240. And then looking across the rest of the image, we're not selecting anything that we shouldn't be. All right, and we'll save this away. Now we do have red in our image just naturally, so I'm going to change the color of our overlay here to purple. That way things don't get confused. Now back to the original image. We can see that there's three different colors here, and we want to decide which one we use for finding our cell. So because we're going to be measuring the mitochondria, and theoretically, based on the experimental manipulations that we use, these mitochondria might be present in different parts of the cell. Perhaps there's some sort of mutation which causes all the mitochondria to ship out to the neurites. So we don't want to have our cell body segmentation relying on the presence of these mitochondria, even though they're right here in this image and it looks really juicy. So when we choose our channels to use, we're not going to use the red channel. So let's start with just the green. So looking at the cytoskeleton, it leaves this big hole in the middle where the nucleus is supposed to be. So how about we add the blue? Now if you noticed, the screen around the outside here got a bit darker. And that's because MIPAR is scaling the whole image so that it can be, so that the max value is shifted down to 255. Right? Because when you're adding two channels together, the end result could be higher than the max number that this image can store, which is 255. And that's, a lot of that is because of the scale bar right here. So let's try to get our green plus blue segmentation done in a way that the scale bar doesn't mess things up. We could try to do some grayscale reconstruction that I talked about earlier, but because our channel operation pulls from the original file, any grayscale manipulations that we use aren't going to be present simply in this operation here. So we'll have to do it manually. We'll take our green here and set it into our companion image, and then we will call, our blue channel and add them together. Okay, that's much better. Our microtubules here, or neurotubules are much brighter. Okay. And so now with this image we can do, let's try our basic threshold. What's going to be a good value? And we can see all the black background here, 1, 2, and then some fuzziness here caused by the JPEG compression. But if we do a value, say, of 3, what does that give us? It looks pretty good, actually. We don't have too much extra selection around the outside, and most all of the PC12 cell is found. Not too happy with that hole being selected. Let's turn our segmentation down, strength down a little bit, 5, 10. When we get to 10, we start to lose some of these for the sake of this. So we go back down to 5. Okay, or even 3. No. Okay. So now we have our basic threshold, and we want to subtract this scale bar from it. Now we overwrote our companion image, so we'll have to switch this around, save into memory image 1. Right, now down here, we will set memory image 2, call back memory image 1, and turn it into our companion. And then call back memory image 2, and minus. Okay, a little bit of selection here around the edge still, but that'll get removed at the same time we clean up this rough selection here on the edges of these. So I'm not too concerned. So now let's do some manipulations here, some cleanup. Say if we start with a closing operation to join this together. So if we dilate uniform 1, 1, a simple one. That looks pretty good, actually, in getting things as close as we want them to be. A road, one. Okay. How about if I rode back 2? No, that starts to look kind of ugly. Just 1. So you'll see that our cell body selection actually extends a little bit beyond the edges of these neurites. And I'm okay with that, because we're not measuring the size of the neurites. Our goal is to capture the mitochondria within them. So if our selection extends a little bit out, that's perfectly fine with me. So now let's do a little bit more cleanup. How about we do some smoothing? It's 5 or 7. 7 looks like a good number here. And then we'll have to do finally a reject small features to pull away this corner here. 24. What else do we have? This 24, 28. And this thing here is 284. So we're going to set 50 as our cutoff. And this little guy here is going to be right. Now we have to fill in holes as well. This tiny hole here, and we don't want to fill this in if possible. So let's do another reject features, this time targeting holes. 9, 129, 15. Again, let's do 20. And here we then have our whole cell selection. We're going to stash this away and use it as our base for finding the neurites and the cell body. Now, the cell body itself is actually pretty straightforward to find. We're simply going to do a strong erosion and remove all these small thin features. So erode, say 25. 25 does pretty good. Here we want to try to have this filled in. See, 30 takes away this selection which we want to have. 25. Okay, and now if we dilate back out, dilate by the same amount, and here's our cell body. So we'll set this cell body. Now let's stash this as our companion. Call back our original memory image 2. And minus. And here we have our neurite selection. A little bit of tiny features here around the edge. So we'll remove these with a reject features. And the basic 20 looks good enough. Well, let's be a little bit more precise. Pull off these tiny little figures, but we don't want this here. Okay. So maybe 150 will be a better number for us. Okay. And this then is our neurites. Now, the last thing to do is find the actual mitochondria itself. So we'll use our red channel to find the mitochondria. And here we run into an issue. We want to measure the amount of mitochondria in the neurites and in the cell bodies. And there's two ways that we can do this. First, we could just measure the amount of red signal present in each of these segmentations. So just sum up all the grayscale values. But I'm not a fan of that specifically because some mitochondria look like they're stained a little bit more dimly than others mitochondria. Maybe they're slightly out of the focus plane. You know, is this bright mitochondria really worth more than this dim one? And that's the decision you're going to have to decide yourself. You know, are you perhaps staining a particular cell surface marker of the mitochondria and you care about the amount of it? In which case, yes, you would measure the grayscale intensity of the red channel that is present in each of these segmentations. In our case, we care about the amount of mitochondria itself, not the amount of staining on those mitochondria. So rather than using the grayscale value, we're going to segment the mitochondria, turn it into a simple yes, no, there is mitochondria in this pixel or not, and then measure that within each of these regions of the cell. So if we do, say, a basic threshold to capture mitochondria regardless of its grayscale intensity, and we achieve something like this. Mitochondria do have a tendency to overlap and be a little bit messy in their localization. Perhaps we drop down to 25. Okay. And now we'll do a reject features to remove all these tiny little single pixels. Yeah, reject features. Okay, and I don't see any red signal that's now not being selected. And I am not going to bother cleaning up the rough edges of this segmentation. It's simply not necessary. We have our best guess of what the mitochondria are here. And this best guess, put out by the microscope itself, and then with a little bit of JPEG compression, does include some of this messy edge. If we were to go in afterwards and change that, just because it doesn't look pretty to us, the cell doesn't care, biology doesn't care. This is the most accurate representation that we have. So we're going to stick with it. So we save this away. Excuse me, the next thing we have to do, of course, is remove this scale bar. So save this away, say in memory image 3, call back our scale bar, which is in 1, set it in our companion, call memory image 3, and minus. Okay, so here's our total mitochondria. And the next step is to divide the mitochondria into two populations. The population that's present in the neurites and the population that's present in the cell body. So we'll save our mitochondria away in memory image 4. Call back our neurites. Right, our neurites here are in memory image 2 because we just set it to memory image 2. So we can call back that, set to companion, recall memory 4, and do an intersection. So now we have all of our mitochondria selection that overlaps the neurites. And this then will allow us to say mitochondria in neurites. So now let's grab the mitochondria that are in the cell body. It's tempting to simply subtract the mitochondria in neurites from the entire mitochondria, but then you end up finding tiny little pieces like this, pieces of mitochondria that are outside the neurite but not within the cell body. So it's better really to take our neurites, or excuse me, our cell body here and do the same thing that we did with the neurites, is do an intersection. Okay. So we'll set our cell body to memory image 5. Now, call memory image 5, set as companion, call memory image 4, and intersect. Now we have a true all the mitochondria that are within the cell body as we defined the cell body earlier. Mitochondria in cell body. Now we have our two selections, here and here. So we want the percentage of mitochondria in the cell body or in the neurite. So we'll measure a percent area fraction, but relative to memory image 4 where the mitochondria are. So now we're going to get the percent of mitochondria. Accept. And if we choose particular layers or two subpopulations of mitochondria, and here we are, 43% in the neurites and 56% in the cell body. And that is our last fast track lesson for now. Next we'll move on into our focus on pre-processing section, where we get really into the depths of all the grayscale manipulations that you can use to pre-process your image. Although I do recommend that you check back to the fast track session from time to time. We might add a couple of more lessons as we discover cool new recipes to show, cool new images, and interesting techniques to describe to you. So I'll see you in our next section.